talk about the SplitsTree app. So I published the first version of SplitsTree in 1998. So it's been a piece of software that I've been working on for a long time now, and there's been many different implementations, and this is the latest one. And I just managed, together with Dave Bryant, we just submitted a paper describing this latest version this week, in fact. And we also wrote a 75-page user manual, which was so painful. I hope that people still read manuals. Otherwise, So I've got a couple of slides. And, so we're talking about computational phylogenetics, and we all know that there's some very difficult problems that people have worked on. Multiple sequence alignment is one of them, where there's very nice tools, but it took a lot of algorithmic engineering, software engineering to get those things to perform as they do. And so a lot of people have put a lot of work into that. And obviously, phylogenetic tree inference methods is another area where a huge amount of work has been done by computer scientists and biologists, and people just working on fast algorithms. So I'm not going to talk about that. I haven't done any kind of work in those directions. So what I've been looking at is more data visualization and exploration, and some phylogenetic inference, more networks and trees though. So it's all in this program that we call SplitsTree app. We didn't want to call it SplitsTree 6 because it sounds like it's just another version of a program that's been around for a long time, but it's actually a completely new program, but designed in a way that hopefully old users will be happy to use the new program. So this program is about it. As the name suggests, it does visualize split networks, but it's also has a lot of features that address the tree visualization, tree comparison, both unrooted trees and rooted trees. And there's also quite a few algorithms for rooted networks. It's something that, in theory, biologists should be using whenever there's reticulation, whenever there's speciation by horizontal gene by hybridization and so on. But in practice, people don't really do because there's no tools that really allow them to explore that idea even. There's nothing that you can just take off the shelf. There's no neighbor joining of rooted networks. So you can't just try something. And so we're hoping to fill that gap. And so what I thought I would do is try and show how you would use this program to look at data. And so I'm not a biologist, so I can't really ask deep and meaningful biological questions. But here's one data set that I've been looking at, which I like. It's a data set of 78 chloroplast genes for 13 water lilies and 4 other plants. So this is the complete set of all protein coding genes in the chloroplast for these 17 plants. And why am I looking at these? Well, any of you who's ever tried to find, who's interested in phylogenetic data and has looked at papers, you know that the papers show you nice trees, but nobody provides the actual data. They say, okay, here are some accessions. Good luck reproducing what we've done here. There's very little actual open data when it comes to phylogenetics. This is one of those papers where everything that was computed, all the intermediate results were actually provided in supplementary material. So that's quite a nice data paper in that sense. And so that's one of the reasons why I'm looking at water lilies here, is because I spent a lot of time looking for data sets, and this is one that I found. And so I want to look at that one a bit. So what do? I get rid of this for a moment, and then I'll fire up the SplitsTree program. Yeah, should. This is taking a long time. There we go. So this is what the program looks like. If you ever use any of the old versions, it looks very similar, but I want to start with this data. So so we're looking at, and I hope you can, is that coming? Yeah, so you can see here, this is one of those 78 gene trees. It's, the name's a bit small, but it's called ACCD, whatever gene that is. And yeah, and so what's interesting, so the question that is being asked in that paper is, are the water lilies monophyletic? And so to study that kind of question, to study that question, what the author did is he put in some outgroup taxa. So these two guys at the bottom are definitely outgroup taxa, and I'm going to mark those. Oh, sorry, not yeah, with mark them red and give them a, I don't know, a diamond. There you go. So we're going to see where those guys fall into the analysis. And these two are also outgroup taxa, but they're closer to the original in-group. So it's one of those studies where we're trying to use outgroup taxa to understand something about the in-group, and everybody else here is an in-group taxon. So I'm just going to, just for us to see what's going on, I'm going to green, maybe a circle. Okay, there we go. Okay, so this is one of those trees, and I routed them using midpoint routing, and so you can see that the really distant outgroup is all the way separated from everybody else. The in-group, the other outgroup, which is closer to the in-group, appears here like as an in-group rather than outgroup. But does that make sense so far? I could, if I wanted, I could, of course, reroute this tree so that the outgroup's on one side. In fact, I'll do that. So I can take all those trees. I'm going to select these 4 taxa here. Okay. And I'm going to reroute the trees, reroute by outgroup. There you go. Now you can see those 4 guys. They're all together in this particular tree, in this gene tree. They all appear separated from the in-group, which is nice. Now we can move along to look at some of the other trees. And this is the second gene tree of the 78. Also looks kind of good. Third. Now this looks kind of, yeah, this is, you can see now that the routing no longer has the 4 outgroup separated from from everybody else. So there is, so what we're trying to look at here is what's going on with the in-group versus the out-group. And so we can look at the trees one by one, and that's going to be pretty painful. So another way to look at the trees is to put them onto a page altogether. So here are all 78 trees, and I guess I should have selected those out-group taxa. Now you can kind of see, I mean, this is maybe too much, so maybe let's do 3 times 4, not all the trees are together, and then maybe you can kind of get an overview already what's going on with the outgroup taxa. Sometimes they all 4 appear as an actual outgroup, but sometimes they are not there. They are separated in the tree. So we can see that there is an issue that these gene trees do not always separate the 4 outgroup from everybody else, and that's the focus of that paper was to try and figure out whether the water lilies are monophyletic or not. And yeah, so another way to look at this data would be, okay, rather than looking at all the trees in the grid like this, we could use a tangogram and look at the trees 2 at a time. So let me try and, there you go. So you can see how these guys look fine, and let's move to the next 2 trees. Also, yeah, sorry. Yeah, this is kind of interesting. You can see there's some major difference in the topology of the trees, but the way that the outgroups are placed seems kind of consistent here. Yeah. Okay, so that's all trees. That's all fine, but we haven't really seen compelling evidence in either way yet. So a thing that you would like to use is consensus methods. So let's not do it this way. Consensus of these trees. This is the majority consensus of those 78 trees. And yeah, a bit hard to see. Sometimes it's better to scale the edges. So here they're by me, in a consensus tree, you take the trees and find out what they have in common, find out what the majority has in common, and then you produce a new tree, and the edges are scaled by the mean length of the input trees. But another way to scale would be by count, which gives you a better idea of what are these trees sharing, if anything. And so now here you can see, here are our 4 outgroup taxa. select those guys. And here are some of the water lilies. And you can see that they are connecting to this unresolved node with the outgroup taxa. So these are the, so they should be over here on the left with the other water lilies, but they're stuck over here. So maybe if we leave the majority consensus and go what's called the greedy consensus, we get this picture, which is slightly better because now the outgroup taxa are separated from everybody else. And we can go one step further. Now this is something that we published last year, which we call a consensus outline. So this is something Banu, who's in the call, and myself, we published. Actually, we developed the concept of phylogenetic outline together with Dave Bryant in New Zealand, which is something between a phylogenetic tree and a phylogenetic network. So for many years, we've been trying to persuade biologists to use networks like this, the so-called split network, where these different parallel lines correspond to the bifurcations in the network, a bit like the edges represent bifurcations in the tree. The drawback with these kind of visualizations is that a biologist would put their finger on one of these nodes in the middle here and try and interpret. They try and take this as an explicit representation of evolution, and they're trying to figure out what's going on here. Why is there 3 parallel lines moving along that way? These are, for us as computer scientists, we know these are just, this is data visualization. For biologists, they're always trying to interpret these things literally. So if we hide all those details and actually using a different algorithm, this is not just hiding details. There's actually a nice algorithm, which we call the outline algorithm, that computes this outline in linear time, linear in the number of splits, or quadratic in the number of taxa. And it's a very straightforward algorithm that I have my students implement in my phylogenetics course. So it's a nice algorithm. In any case, well, what we can see here now is, here are those 4. There's those water lilies that should be over those guys, and we can see, well, there is some of the trees are putting the 4 outgroup taxa, separating them from everybody else. And this is what these 2 parallel lines represent. But there's some of the trees, nearly half the trees, well, nearly as many trees, put the closer outgroup with these taxa here. And so, so this phylogenetic outline kind of confirms what the author wrote in the paper, that you can actually figure this out. So the chloroplast genes on their own do not clearly answer the question whether these water lilies are monophyletic or not. So more work needs to be done to solve this. But the main goal here was to give you some idea of what we're trying to provide here is a tool that allows you to explore your trees, explore your data. Yeah. So that's, okay, let me get back to the slides for a moment. So, yeah. So the second thing that I wanted to look at, this is also something that I did together with Banu. We looked at some data from a language database. So phylogenetics, usually we look at biological data, obviously, but tools that have been developed in phylogeny are also used to some degree in linguistics when people are trying to study the evolution of languages. And there the data comes in the form of alignments, and these alignments usually binary data, and the binary data, each column in such an alignment represents a cognate word, a word that has the same meaning. And then basically, and then you have, if that language has that word for that meaning, then there's a 1, and if it doesn't have that word for that meaning, it's a 0. That kind of data. So you have binary sequences, and there's a nice database that Simon Greenhill has put together, which contains languages from New Guinea, which has a very large number of different languages. So it harbors a substantial amount of the languages on Earth. And yeah, so here we looked at a data set of 13 languages and run them through BEAST. BEAST is a algorithm, how that, it's an MC algorithm that takes an alignment and then it samples from the posterior distribution of the trees, tries to produce a profile of trees that represent the probability distribution of trees given that data. And so let's look at that in this program. Sorry, when do I have that? Here. Is that the right one? That's actually not the right one. Sorry. I should have. Here. Yeah, this one here. Okay, so now if you ever run Bayesian analysis, you know that you get a profile, usually by default 10,000 trees or actually 10,001, or in this case, we've got 30,001. The one, I guess, is the first one is always the starting tree, and then you get the ones that get sampled by the MC algorithm. And what we're particularly interested in is this language here that I'm going to highlight. I don't know, give it a background color of orange. Okay, because this it belongs to one group of languages. Is these guys? It really belongs with these three guys here, but you can see these three from the same language group up here, but you can see already in this tree, it's actually placed over with these three languages. Okay, so it's a typical situation that we know, we would think of as horizontal gene transfer, maybe that parts of the language from one language group are being transformed into the other one. And so depending on what you're looking at in terms of vocabulary, sometimes you go with the one, sometimes you fit with the other. And if you do a Bayesian analysis, then you would capture some of that. And so one tool that people have developed in the Bayesian phylogenetics world is a so-called density tree. So this is the idea here is that you draw all those 30,000 trees on top of each other. And I like to actually use this layout, but it's kind of slow to draw. And so what you see here is in orange, if you can see those orange lines are a bit thin, is a greedy consensus tree. And then what you see in black and red are all those Bayesian sample trees. And what we're doing here is drawing any part of a sample tree black if it corresponds to the consensus tree. And we're using red if it is in conflict with the consensus tree. And so the amount of red smudge that you can see there gives you some idea of how many times in the Bayesian sampling you're obtaining something that differs from the consensus. Yeah, I'm going to go back to the triangle tree because that's kind of faster to compute. And so, okay, that's one way that people like to look at things. And now we're going to again look at the consensus. We want to do some work with consensus splits. And what we want to do is we want to have these side by side. So, yeah, so on the one hand, and we're going to try and, first of all, you can see the orange. Let me just, the orange taxon here that I mentioned earlier, you can see in the density tree visualization, that there is, some of the trees are putting this taxon with the phi below, and most of the trees are putting with the phi above. So that's that language that has some borrowing going on between the two different families. And now over here, this is just a mundane majority tree. And, we don't see that here. There's no evidence in a tree, a majority. So in a way, this is a kind of a slight critique. We're criticizing consensus trees. At least think about any kind of tree visualization is, you have to choose. It's a linear size visualization. So you're making a lot of choices, and what you finally end up showing is hiding a lot of stuff. So here we're showing one the consensus tree here. And so if we were to go to the greedy consensus, I don't think it's going to look very much different. Yeah, now if we go to let's go to the consensus outline. Yeah, now we do see something here. I'm going to scale by count again, rather than by average length. And so now we're seeing something that's a bit more interesting. It's got this ring. So this is now a split network. This is a network that's based on the Bayesian profile. And so one obvious difference between the density tree and the network is that the density tree is rooted in the network. It's not, well, that's not something that we can. I mean, we can root a network, so we can just say let's do midpoint routing. There we go, and then we've got something that's looking very similar. Now, what you're seeing here is like this guy, obviously in the rooted consensus outline, is conflicted in placement, and we see this here clearly in the density tree on the left. And our other candidate who we were looking into is also obviously conflicted here. I can actually try and clean this picture up a bit. Yeah, any case. So that's just to, again, my main message here is what we've tried to do with this program is integrate a bunch of different methods to make it possible for biologists to explore both trees and networks together. So I guess, how many, have I got about 5 minutes left, or is that, or 3 minutes?
Sure, around 3 minutes.
Okay,
3 to 5 minutes.
Then I'll just go back to one more thing, a final thing here. Going back to these, so I'm going back to that data with those, the water lily data. Now, what I want to do here is I don't want to use all the trees, so I'm going to take a subset. I'm going to take the trees and then filter them. Somewhere there's a filter, tree filter. Okay, I'm going to, for now, I just want to take 2 of the trees. I want to take the first 2 trees. And what do I want to use them for? I want to briefly show a method, one of our methods that we've implemented for rooted networks. Okay, no, I'm going to do it differently. Here we go. There we go. So this is a so-called hybridization network. It's a network that contains both of those input trees that I selected. And it's, they're rooted trees. So this is also a rooted network. And it has minimum hybridization number. So among all networks that contain those 2 trees, this is one of the ones. And there's actually 3 different ones in this case that is minimal with respect to a hybridization number. So that's an algorithm that we've had around for a long time. It takes 2 trees as input. They can be multiplicating with unequal taxon set. And what's new is we now have an algorithm where you can take more than 2 trees and filter those, I guess. The autumn algorithms, I don't know, it's still going to run on 2. But what we can do with those trees is run an algorithm called the ALT algorithm. And now that's a network that contains all those 4 trees. This is heuristic, so I'm not 100% convinced that 11 is the smallest number of hybridization nodes possible. But the hope here is that we're trying to provide algorithms where biologists can take a set of gene trees and then explore the idea of embedding them into a network. Yeah. Well, I've got this here. One final thing, as I mentioned, the frustration that anybody who's ever tried to find phylogenetic data in papers has, we've tried to come up with something to address that. Namely, we've implemented a QR code. Now, this QR code is not a link to a web page. This is actually is simply the Newegg description of this particular network here. So if you took a photo of that, you would have a Newegg string, and you can just paste it into your favorite tree drawing program. And so we have that for trees, for routing networks, and but also, and this is new for split networks as well. So, yeah. So if you just might show you one final slide. If you were to download the SplitsTree iOS app, it's a bit dated. This is a test version. There will be an update very soon. Then you could actually be doing this on your phone or your iPad. Yeah, you could capture. Yeah, so I'm moving forward. I hope that people might put QR codes onto their trees and networks and make it easier to share data. Yeah, thank you.